Okay. Well, good morning. We've been talking about the basic structure, nomenclature, classification, replication cycle of viruses, and now I'm going to talk into more detail about the replication cycle of certain DNA viruses. But before I go on, are there any questions about any of the other stuff? Okay. Okay, well, today I'm going to start some lectures on the life cycles of certain DNA viruses. Now, we have a lot of DNA viruses, and I can't go into detail about every DNA virus there is, so I'm going to focus on the ones that cause human disease, and I'm also going to focus on ones that illustrate some of the typical things that viruses do, so that you see it in context of a particular virus, but some of the concepts about how viruses go about doing things are applied from one virus to another, because they face certain problems, and I'm going to try and point that out as we go through. There are only a limited number of solutions to those problems. So weird and wonderful as some of these viruses are, it's just that they're using a series of solutions. So in a way, you can think of it as a series of modules, how you correspond to each particular challenge that viruses meet. And the way they put these modules together for a particular virus may be unique, but the ideas behind those modules are common to many of them. So we're going to try and extract some basic principles at the same time as we talk about particular viruses. So first of all, to try and extract some basic principles, I want to just try and get an overview of what we mean by viral strategy, and we'll keep coming back to some of these points. One thing is, not that a virus has got thoughts, but if you want to put thoughts into a virus, its main aim is to copy itself, get out of the cell that it's replicating in, and get into another cell. And that basically is kind of the virus's aim in life, if you could talk about such a thing, which of course is not scientific. What viruses need to do is they need to ensure that they replicate their genome, because without that they're not going anywhere. And they need to ensure that that genome is packaged, because, as we said, in order to get into the next cell, they're going to need their genome packaged in some kind of thing which has an attachment protein on the outside so it can recognize the receptor and get into the next cell. It also needs to protect the genome while it's out in the big wide world. So you've got to at least ensure that it's packaged for protection purposes, and also that it gets an attachment protein so that it gets in via the receptor on the next host cell. And in doing so, it will alter the host cell to some degree or other. Now, the simplest thing is all you do is you alter the host cell, because the host cell is now making your proteins as well as its own proteins. But many of these viruses will change the host cell in some way or other to make it a better environment for them to replicate. So this is what we mean by strategy: how they replicate their genome, how they make sure they get out and get packaged, and what they do to the host cell. Now, much of what they do to the host cell is very complex and detailed, and I'm not really going to be discussing it in these lectures. We're going to focus on just how the viruses replicate. But of course, much of what they do to the host cell is what eventually leads to the disease. And we can't discuss the molecular basis of pathogenesis for all of the diseases, but a lot of it, as I say, is almost side product of what the viruses do in order to achieve their strategy. Okay, so, and as I say, strategy is just the way that's done. But when we talk about strategies, what we're always looking for are the weak points, the points at which the virus is vulnerable to us trying to inactivate it in some way or other. So clearly, one of those weak points is whenever a virus does something for itself. Another weak point is if it uses some of our host machinery that we can temporarily do without, so that if we take something to inhibit, we survive because we can do without it for a few hours, but the virus can't. So we're looking for these kinds of things. We're also looking for how the virus can evade our defenses, and many of these viruses have got ways to evade our defenses. And so if we understand those, maybe we can get at them that way as well, by preventing them invading them. So we're going to be looking out for a lot of these weak points. This is just, again, some overall concepts that are always behind all these strategies. One is the host cell has got certain pathways that exist, certain rules that are used in order that everything in the host cell is targeted to the right place at the right time and does the right thing in the right place. And viruses have to live by those rules. You know, you have certain signals on your messages which tell the ribosome this is a message. So if viruses are providing messages, they either have to obey the rules and put the same features on their RNAs to say that their RNAs are messages, or else if they're going to break the rules in some way, they have to make sure that the host cell protein translation machinery still recognizes these atypical messages. And that can actually be just as much a problem as going by the rules. So a lot of what viruses do are they are trying to parasitize the host cell. They have to provide the same signals that the host cell will provide. So some of this is just a consequence of trying to mimic the host cell, even though they're rather different types of things. And so this is just to remind me that this is a cell which has been dually infected actually with two viruses, a SV40, which is growing in the nucleus. That's a DNA virus. It has icosahedral nuclear capsids, and all these little black dots in here are icosahedral nuclear capsids. This is an electron micrograph, of course. And here is the cytoplasm, and it's chock full of what look like little bits of unraveled knitting wool, which is a paramyxovirus, which has a helical nuclear capsid. In this case, it's an RNA virus, not that it matters at the moment, but it's an RNA virus with a helical nuclear capsid. And part of my point is just to point out the cytoplasm here, if you can see it on your laptops, is absolutely chock full of these helical nuclear capsids. The nucleus has got a lot of these icosahedral nuclear capsids. But if you look, the SV40 is in the nucleus, the paramyxovirus is in the cytoplasm. These viruses don't just sort of take over the cell randomly. They tend to, if they're a cytoplasmic virus, they use this area. If they grow in the nucleus, then as we'll see with SV40, which we're going to be talking about today, they replicate in the nucleus and not in the cytoplasm. So the location of where these capsids are assembled is very typical for each virus, but in order to get into the nucleus, it has to use nuclear localization signals, just like our proteins do. So a lot of what we're going to be looking at is how viruses manage to obey the rules despite the fact that they're not they're just small parasites, or how they can bend the rules. So I'm going to start off by talking about DNA viruses, and bear with me till we get to the RNA viruses, but having DNA means that you use fairly different strategy than having RNA, for what I hope will be obvious reasons when you've gone through all this. So anyway, let's focus on the DNA viruses today. They have to make messenger RNAs to make their own proteins, so somehow they've got to make messenger RNAs. Now, we make messenger RNAs ourselves, of course, using polymerase II, and polymerase II uses DNA as a template and makes RNA. So it's a DNA-dependent RNA polymerase. And also, if you're going to make messenger RNAs, our messenger RNAs at the 5' end have got a GPP cap. That is methylated, so they have a methylated cap structure at the 5' end. That's involved in the ribosomes recognizing them as being genuine messages. It's also involved in keeping them stable in the cell. And they have poly-A at the 3' end. And that is also involved in translation in a fairly indirect way, but it's also involved in keeping them stable. So our messages have got these features at each end which are involved in stability. They're also involved in the ribosomes recognizing them as efficient templates for translation. So we also have those proteins, the enzymes that are needed to add those to make a properly modified messenger RNA. So we have all that, obviously, in our cells. DNA viruses also need to replicate their DNA, and of course we replicate our DNA, so we have a DNA polymerase, plus the additional proteins that you need to replicate DNA, all those things for making Okazaki fragments, for removing the RNA primers, for ligating it, the histones. We have all of this stuff. So we have the machinery that you would need to make messenger RNAs off a DNA template. We have the machinery that you would need to replicate DNA. So for DNA viruses, we actually have a lot of this machinery. But of course, for us, that's in the nucleus, because that's where we copy our DNA into RNA, and that's where we replicate our DNA. So all this machinery that we have is in the nucleus. But basically, this is a treasure trove for a DNA virus because since it's got DNA genetic material, it could just use our RNA polymerase and our modification enzymes to make its messages. It could use our DNA polymerase and our DNA polymerase associated machinery to replicate its DNA. But since that machinery is in the nucleus, if it's going to use it, it's going to go into the nucleus. So SV40, as I said on that previous slide, was a DNA virus. I'm not at all surprised to see it's in the nucleus. But you can get DNA viruses which replicate in the cytoplasm. But if you're awake enough to think about it at this time in the morning, if it's going to be in the cytoplasm and all this machinery is in the nucleus, then if you're a cytoplasmic DNA virus, you're going to need to code for cytoplasmic DNA and RNA polymerases because our polymerases all got local nuclearization signals. So as soon as they're made on the ribosomes, they go off into the nucleus. So either you've got to find some way to keep them in the cytoplasm or you have to make your own enzymes. And the solution, the cytoplasmic DNA viruses that we're concerned about is they make their own enzymes. So they need to code for cytoplasmic DNA and RNA polymerases. And, of course, they'll need to code for the accessory proteins needed for DNA and RNA synthesis because, as I pointed out, it's not just as simple as having the polymerase. Of course, you've got all that accessory machinery that you've learnt about in biochemistry. So this is actually a lot of proteins that you need to do to replicate DNA and RNA. You need a lot of proteins as part of your genome in this case. So for these viruses, you can predict they've got large genomes before you've even heard anything about them. If you just think about the basic properties of what you would need if you're a DNA virus. If you're going to the nucleus, you can take advantage of a lot of what we provide. If you're in the cytoplasm, you have to provide it yourself. That's a considerable amount of genetic material. So the larger the cytoplasmic viruses are going to be amongst the larger viruses. And the pox virus family, the true pox viruses, are considerably larger than the smallest of the nuclear DNA viruses because, as I say, they've got to provide an awful lot for themselves. Okay, so here's a slide to give you some idea of the range in size of the DNA genomes of the DNA viruses. And here, if you look at the smallest ones, they're about 5,000 kb as a kilobase, so 5,000 base pairs, compared with the pox viruses, which are getting as almost up to 400,000 base pairs. E. coli is much bigger than that. Don't learn these unless you're just fascinated by figures or want to wow everybody at virology trivia questions. I'm giving these so that you have some idea of the relative sizes, just so that you feel where we're going. And we're going to start off by talking about the smaller ones, and we're going to work up in size to the larger ones. But I'm just giving you the sizes. And you'll notice there in your notes, I haven't used to bother to give them, and then people sort of said, could I put them in? So I put them in. So just blame previous M2 classes. Okay, so we're going to start off by talking about these small DNA viruses go up to the larger ones. So parvo, as I say, means small. So the smallest DNA viruses that we're going to be talking about is just the parvoviruses, and I'm going to say very little about them here. I just want to give you some idea of what they are. They will reappear briefly in some of the clinical virology. But parvo means small, and in fact they have single-strand DNA, and I'm not going to talk anything about how they replicate it with about 5,000 nucleotides to the genome, as I say. Now, 5,000 nucleotides is not enough to code for an RNA polymerase, a DNA polymerase, and all those accessory proteins. So if you know that this is 5,000 nucleotides, or you know it's the smallest of the DNA viruses, you know it's going to go to the nucleus, because that's the only place it's going to be able to find all this machinery that it needs to make its messages and to replicate itself. So these replicate their DNA in the nucleus. And one of the consequences of replicating your DNA in the nucleus, which is going to come back multiple times when we're talking about DNA viruses, is we only make the machinery for DNA replication when our cells are about to divide. So when they go into S phase, they start making all the things that they need for DNA replication. The DNA polymerases, the accessory proteins, the histones, The nucleotides, we make ribonucleotides all the time. We need to keep those to make our RNA to keep the cell functioning. But the deoxyribonucleotides, we only make during the S phase because we don't need DNA precursors until we go into S phase. So it's a waste of time and energy to make those unless you're about to divide. So all these things that you need to get so that you're ready to make DNA and do it efficiently and quickly and smoothly and easily are done in S phase. So at the beginning of the S phase, all these things are switched on, and then as these precursors accumulate and all the enzymes accumulate, we replicate our DNA, and then eventually we'll go into mitosis. So if you infect cells which are not dividing, they're not going to have those reagents available for making your DNA. So DNA viruses have to either go into cells which are already dividing, if they're going to take advantage of the DNA synthesis machinery, or they have to cause cells to divide. Parvoviruses go into cells that are already dividing. There are animal parvoviruses which fortunately don't affect us. They replicate in the gut because you've got plenty of dividing cells in the gut. The human parvovirus that is the current one that we're aware of, there may be others that have yet to be found, but the one that we're aware of and we will come back to talk about briefly, infects dividing cells in the bone marrow, predominantly erythroid precursors. So in fact, when you're infected with this virus, in most people you get it as a child, and it's a relatively trivial disease called fifth disease, actually, is the old word for it, and we'll come back to it. So when I'm talking about clinical stuff here, it's only to put you in the picture, so as we go through these viruses, you know what they're associated with. We'll come back and discuss the clinical picture more later on. But what this does is, since it grows in erythroid precursors, you get a temporary failure in red blood cell production because the cells are diverted to viral replication rather than to making red blood cells. For most of us, that's short, and our red blood cells are relatively stable, so it isn't a big problem. It's a fairly minor disease. But for anybody who suffers from any kind of chronic hemolytic anemia, where the blood cells turn over very quickly, then you stand the chance of anemia and even, in serious cases, an aplastic crisis, because if your blood cells don't survive this period when you're not making new fresh blood cells, then you run out of red blood cells. So this can be a serious problem in people with some kind of hemolytic anemia, including sickle cell thalassemia, and it can be a cause of aplastic crisis. So this is one of these viruses that you normally is not a problem, but if you know that you have a patient with some kind of hemolytic anemia, it can be a real problem. And they have icosahedral symmetry, so they're little icosahedral viruses, pretty simple, replicate in the nucleus, and you don't need to know much more about how they do it. So let's go not much up in size. These basically about the same genome size. Another couple of families which we're going to talk about a bit more, the papillomaviruses, which cause warts, and some of the warts are benign, and some of them can lead to cancer. And cervical warts, again, some are caused by benign members of this family, and others are caused by members of this family which seem to be very important in the progression to cervical cancer. So they seem to be necessary but not sufficient. So getting one of the strains that causes progression to cancer doesn't mean you'll get the cancer, but it certainly increases your risk factor. These are small, naked icosahedral viruses again. So again, being that small, you can bet that they're going to go to the nucleus. They have an icosahedral capsid. Here I put it's mainly VP1 with some VP2 and some VP3. I'm not stressing that you should know VP1 and VP2, VP3 and what they do in the nucleic capsid, but VP means it's a virion protein. So VP is a way to talk. If you remember, I said any protein that's in the virus particle is a structural protein. VP means that it's structural protein. So if you see something labeled as VP, you know it's going to be part of the virus particle. Non-enveloped. Circular DNA. And in this case, it's packaged with histones. So it's inside the nuclear capsid, but it's packaged just like our DNA is packaged, and it's packaged with our proteins. So this is a virus that uses our proteins to package the DNA, and then it puts the DNA inside this protective nuclear capsid, which of course will have the attachment protein on the outside. The papillomaviruses have got a slightly larger genome than the polyomaviruses. Don't worry about that. At one stage, these were put together in the same family because they package the same way. They both got circular DNA. They look very similar. And once we began to look at the life cycle, we realized that the papillomaviruses have got a very different life cycle from the polyomaviruses. And I'm not going to talk about the papillomaviruses today. I'm just going to talk about the polyomaviruses. As I said, the papillomaviruses cause warts, and some of them are associated with cancer, not just cervical cancer, but some other cancers they're associated with. But the virus itself is never sufficient for the cancer. It may be a necessary step in the cancer, but it's never sufficient. All these cancers are multi-step things. But we're going to be talking about the polyomaviruses, partly because they were the first viruses studied in real depth. And the ones that were studied were mainly ones in mouse. And in mouse, these viruses tend to cause a lot of tumours. Poly means a lot, and oma means tumours. Fortunately, in humans, because we do get infected by these viruses, they seem not to be associated with cancers in humans, but they are in rodents. In humans, the disease that they're associated with, there's several diseases they're associated with, and we'll be talking about those briefly later on in the clinical section. But one of the diseases is a disease called progressive multifocal leukoencephalopathy, or PML. And it's a disease of the white matter, and it turns it into mush, basically, and it's a very nasty progressive neurodegenerative disease. And we'll talk about that later on. But I'm just pointing out these do cause various diseases in humans. SV40 infects humans, BK and JC infect humans, and we'll be talking more about these. These are viruses that are associated with renal disease and with PML. And this is the virus capsid. This is a X-ray crystallograph of the virus capsid. And for those people who are interested in penton spotting or hexon spotting, here's a penton with 5 neighbors, and here's a hexon with 6 neighbors. So it consists of 12 pentons plus hexons. And in its life cycle, it has a lytic replicative cycle where the virus infects the cell, more virus particles are going to be produced, and that's what I'm going to be talking about now. It also has a non-lytic cycle where it can become integrated into the host cell, and I'm not going to be talking about that. I think Dr. Richard Hunt will be talking briefly about that. But we're going to be talking about the productive cycles of these viruses. Okay, so SV40 has a naked nuclear capsid, binds to the outside of the cell. The attachment protein on the outside recognizes the receptor, taken up into an endosome. In the endosome, it becomes acidified, and somehow, once it becomes acidified, it can get out of the endosome, we don't know how, into the cytoplasm. And then it appears that it takes a ride on the host cell machinery, the microtubules probably in this case. It's got the right signals to be recognized as something to take to the nucleus. And it's taken to the nucleus, and it seems that what happens is the nuclear capsid itself doesn't go through the nuclear pore. It seems you can find empty nuclear capsids sitting by the nuclear pore, and presumably they have allowed, their DNA has been released and enters the nucleus. And as I said, it's packaged with histones. So now you've got a double-stranded DNA packaged with histones. And it looks pretty much like regular DNA as far as the host cell is concerned. So it's now in the right place to be copied by the host cell RNA polymerase. So fairly simple. As I say, we're talking about the lytic cycle. And again, I need to introduce some terminology. In the lytic cycle, we see an early phase. And by definition, for these DNA viruses, the early phase is what happens before DNA replication. Late phase starts with the onset of DNA replication. So what happens in the early phase, and in general this applies to all of these viruses, They make any proteins they need for DNA replication. So very frequently, they may not be making all of the machinery, but they make a bit of the machinery, and we'll see what they make as we go through each individual virus. They often make regulatory proteins which change the expression of genes in the host cell to their advantage for what they need to do. And again, we'll be seeing examples of that. And they often make proteins which alter the host cell and result in an environment which is more conducive to replication of the virus particle. And again, we'll see some examples of that. The late phase, as I say, by definition begins with the onset of DNA replication. So by definition, that's when you're going to see the DNA replication. And the structural proteins tend to be made in the late phase as a rule. You don't bother to make too much in the early phase, because if you think about it, at this stage, you've only got one DNA template in the cell. You've infected it with one virus particle. You've got one DNA double-stranded circle in the nucleus at this stage with SV40. You don't have very much template to make a lot of structural proteins. You're going to produce tens of thousands of virus particles, possibly. There's certainly thousands. And so you're going to need a lot of proteins. And to do that, you're going to need a lot of DNA templates. So you don't normally start to do that until you've got into the late phase. So early phase is basically setting the scene, making sure that everything's in place, that DNA replication, once it starts, will go smoothly and efficiently. So what happens with SV40? And as I say, remember at this stage we've got one DNA molecule in the nucleus. And that shouldn't be there yet, but it's decided to put in appearance early. Okay, so what happens? This gets into the nucleus, and there is a perfectly good promoter here, which is recognized by the host cell as being a perfectly good promoter. It actually also has an enhancer, which is a really good enhancer, and turns on the host RNA polymerase very heavily. So it copies a lot of RNAs from this promoter. Again, don't worry about the numbers. This is just the way that the genome has been numbered, and it makes it useful for me because I can tell you where you are, so you don't need to raise your heads and actually look at what I'm trying to point out if you don't want to. But at about map position about 69 here, there is this strong promoter with a strong enhancer, and the RNA polymerase 2 recognizes this very well and produces a lot of copies of an RNA which goes almost halfway around this circle, and it's copied in an anti-clockwise direction, and it has a capped methylated 5' end and a polyadenylated 3' end, which are put on by the host RNA enzymes that cap and methylate and polyadenylate. All of this is available in the nucleus. It's taking advantage of all of it. So all it's done is it's provided a good promoter sequence with a good enhancer to make sure it gets a lot of RNA off it. This is called the primary transcript because this is what polymerase 2 actually makes. But just like so many host cell messages, this isn't actually what gets to the cytoplasm. It has to have some introns removed. So in this case, there's an intron that is removed here. So this is the exon. This will be actually removed and spliced to this. But we know the sequence. There's a start codon here. There's a stop codon here. So the rest of the RNA will not be translated. It just makes this relatively small protein. So this will now be transported to the cytoplasm because, of course, all protein synthesis takes place in the cytoplasm. And again, it will be transported by what takes our messages to the cytoplasm. It's just parasitizing everything that we do. And this is translated into something called the small t antigen. The T comes because, as I say, these were originally looked at in mice, and these were antigens that were associated with the tumors that they caused. And so hence it gets T for tumor. And I'm not going to be talking about this anymore. It's not going to play a major role in the lytic cycle. It's more important in the transformation of the cells, and I'm not going to be talking about it anymore. But there's an alternative splice, so you can take out this intron and you make the small t antigen, but you can take out a bigger intron here, and when you do that and splice the two sides together, you've taken out the stop codon here. This black arrowhead shows the stop codon. So you've taken out the stop codon from this one, and so now it starts translation here. It translates through here, and when this is spliced onto here, this is still in an open reading frame, and it translates a protein that goes all the way around here, and it doesn't meet a stop codon until you're right up here, about 15 on the map position. So this makes a much larger protein, and with enormous degree of originality, this was called the large T antigen, which is really annoying because it means I can't talk about T antigens. You won't know which one I'm talking about, so I have to call it the large T antigen. So this plays a really important role in the replication of the cycle. Remember, I said in early stages, you make the proteins that you need to get going on the late stages, and T antigen is necessary for that. So this is really simple. There's a single early RNA, a primary transcript, that can either have this intron removed, in which case it goes to the cytoplasm and copies of the small T antigen are made from it, or it can have the larger intron removed, in which case the RNA goes to the cytoplasm and is translated into many copies of the large T antigen. This has got a nuclear localization signal. It comes back into the nucleus because it's going to be needed in the nucleus in order for DNA replication to occur. Okay, so we now go into DNA replication. So setting the stage was fairly simple in this case. Okay, so what we now have is the DNA and the large T antigen, which is this little pink oval here, and it binds at about map position 69, which is where that promoter and enhancer for the early RNA synthesis were. But it binds just a bit upstream from that, and in that same upstream region from the early RNA. It binds here, and in doing so, it enables our host cell DNA polymerase to recognize this as an origin of replication. If you remember back to DNA synthesis starts at an origin of replication. And we have a limited number of origin of replications on our chromosomes so that when DNA synthesis occurs, it doesn't just occur randomly. It starts at origins of replication and then spreads away from them. So when this DNA goes into the cell, it's not seen as having an origin of replication because there needs to be something there that says to the cell, this is an origin of replication. The cell doesn't just recognize it as sequence. It needs something else. And the virus provides that with this large T antigen. So once the large T antigen binds here, then the rest of the host cell DNA polymerase synthesis machinery recognizes this as an origin of replication and will bind here and will start replicating the DNA. So this is the start of the late phase. And this is replicated by the same mechanism that you have talked about before. So it has a leading and it has a replication fork. The replication fork will go around the circle in both ways. It has a leading and lagging strand. There are RNA primers. It makes the Okazaki fragments. The RNA primers are removed. The gaps are filled in. The bits are ligated on the lagging strand, just the way that they are with our host cell, because it's being done by our host cell machinery. So it basically, all it's doing is providing the signal that says this is an origin of replication. Our machinery says, oh wow, here's an origin of replication, let me replicate you, and does the rest of it. So this is replicated using host cell replication machinery, and as the new DNA is made, it's coated with the host cell histones. So we provide an awful lot for this virus. So you've got one molecule, it's copied into two molecules, and then those two molecules are each copied. So now you've got 4, so you go from 1 to 2 to 4. The 4 are each copied, now you go to 8. And if you work it out, if you were to copy these like every minute, after 1 minute you've got 2 copies, after 2 minutes you've got 4, 8, 16, 32, 64. The power of doubling every minute means that in 20 minutes you would have about 1 million copies. In 30 minutes you'd have 1 billion copies. So the power of always increasing things by 2 means that in a very short time you've got a huge number of copies of this DNA in the nucleus. If you ever meet anybody who offers you $1 one day and says, I'll double it every day, will you accept it? Say yes, because within 3 weeks you'll be a millionaire. This is the power of that copy. So what we have, I said, that's why you don't bother to make structural proteins of one DNA template at the beginning. Because once you start DNA replication, in an hour or less, you have got so many copies that you can now make huge numbers of RNAs and really swamp out the cell and make all your structural proteins in one big go. And that's an advantage, because if you start making them early, you start giving the cells clues that they're virally infected. And really, the idea is you want to stay under the radar screen till you're ready to go and really go full tilt and do everything you want. It's a kind of a terrorist cell. You know, it lies quiet until it's ready to really take off. So you get all these new copies of the DNA, and now you can go and make your late messages. So before I start on the late messages, are there any problems? There's questions. The small T antigen I'm not talking about. It appears to play more of a role in the transformation during tumor formation, and I'm not going to discuss it in these lectures anymore because I'm just focusing on the replication cycle. These extra antigens appear to play more of a role in those the tumor transformation of cells side of the properties of these viruses. And in these lectures, I'm talking about basic replication cycles, not the transformation. You'll be getting lectures on viral transformation from Dr. Richard Hunt later on. So we now need to make our late proteins, because you've got to make those viral capsid proteins, and you've got to make lots of them, because if you're going to make lots of virus particles, you're going to need lots of proteins to assemble them from. So the late T antigen, as we say, is bound in this promoter enhancer region up here at about 69. What it does is it actually suppresses early RNA synthesis by POL1. It actually acts as an inhibitor of POL1 going this way to make early RNAs, but it really turns on POL1 to go this way to make late RNAs. So in a way, here it's acting as a transcription factor and turning on the late promoter. So it's needed for the RNA polymerase to recognize the late promoter and to stop bothering about the early promoter much. So again, you make a single primary transcript, which is capped and polyadenylated by the host cell machinery. And again, this is not what makes it to the cytoplasm. What makes it to the cytoplasm are spliced RNAs, and our splicing machinery is in the nucleus. So that's good for these DNA viruses. They're using host cell RNA polymerase, capping methylation, and splicing enzymes. They're all there in the nucleus. So you begin to see why this virus is going to the nucleus. And the histones are in the nucleus. Everything's there. So this RNA is alternatively spliced. This little intron is taken out here, and the resulting splice message, so the dot bit indicates where it's taken out. The resulting splice message codes for this protein called BP2. So what I've shown here is the coding region of the RNA is shown by this broad arrow, which I'm afraid is not showing up too well here, but I think it's probably better on your handout sheet. Or a larger intron can be spliced out, in which case you make a protein which has actually got the same sequence as BP2 at this end, but starts later. So it's missing part of its end amino terminal end, and that's called BP3. These both end at the same stop codon. They both got the same carboxyl terminal, but their amino terminal is different. And these are components of the viral capsid. But the major component of the viral capsid is called BP1, and that has an even larger intron spliced out. And it starts here at the beginning of this cross-hatched region. That's where its start codon is. And then it is translated from here round to this black arrow here. And this one is the major protein that's produced and the major protein in the nucleic capsid. So why do I have this cross-hatched region here? And let's look at that in detail on the next slide. So here, if we look at that arrow, that corresponds with the sequence here. So here is the sequence that we see in that region that is shown here. And when we look at VP1, there's a start codon here, AUG. I know that AUG is the start codon because I remember it's in the word inaugurate. So AUG is in the word inaugurate, which is why it's about the only codon I really am very good at remembering. So here, it starts at the AUG. The polymerase, the ribosomes will bind to the message. They'll scan along from the cap until they find a good start codon. And when they find the start codon, they will start translating this message. And so it will code, if we read AUG, GCC is alanine, CCA is proline, ACA is threonine, AAA is lysine. Don't learn this, by the way. I didn't expect you to, but in case anybody's neurotic. But here, VP3 and VP2, their ribosomes are already translating a protein as it comes up to this little purple region. And they are translating in a different reading frame. So they're reading GAU here, which is aspartate, and then GGC, which is glycine. And so they're reading a different region here. And this is something you see quite a lot with viruses, and that is they've got a very small genome. So how are they going to make a lot of proteins from a very small genome? One thing is to use alternative splicing so that you make multiple messages which code for different proteins. But also, you can actually use the same sequence to code for more than one protein because there are different reading frames. And this is a theme that you find coming up time and time again in the viruses, the use of alternative splicing, the use of overlapping reading frames in order to get more bang for the buck, as it were. Now, we, of course, use alternative splicing as well, and it turns out we also use overlapping reading frames. But we don't use it as frequently as the viruses because in many cases we don't have the pressure to do it. But also because in many cases, because we have such a large genome, people haven't thought to look for these kinds of tricks. And when they find, and they're beginning to find them, as the Human Genome Project is forcing us to look in much more detail at what we do. So a lot of what was thought to be sort of special features of viruses because they had small genomes, turns out, as I say, they haven't had any kinds of originality, really. We do these same kind of things, we just don't do it as frequently. So here, the initial RNA has been alternatively spliced to give two proteins which are similar, but one is just starting at a later point, and so it's got less of the amino terminal than the other. And this protein is completely different from these. And it even in one place, as I say, in one region here, the same coding, the same RNA sequence is actually contributing to two very different proteins. Any problems? Okay. So what are the roles of the large T antigen? This is another thing that we see in viruses, and that you free a lot of multifunctional proteins. And again, that was thought to be a special feature of viruses. They're not making many proteins, so you expect each protein to have to do a lot. And with eukaryotic, with our proteins, when people found a protein and they found what it did, they tended to say, oh, well, you know, we got a large genome, it does so-and-so, that's what it does. And now, again, we're beginning to realize that, no, we've got a lot of multifunctional proteins which do very different things in different places and at different times. So we use the same thing again. But viruses, it's a very common feature. So if you take this large T antigen, it's involved in DNA replication. It's involved in increased transcription of late genes because it enables the RNA polymerase to recognize that late promoter. It decreases transcription of the early genes. You don't want to keep on making T antigen once you've replicated your DNA. So it decreases transcription of early genes. It causes cellular transformation, and I really not going to talk about this too much, but just to say briefly, we've said that this is going to use the host cell synthetic machinery. I said the host cell synthetic machinery is only turned on in dividing cells. So SV40 would either have to take the parvovirus attitude and replicate in dividing cells, or it could turn on cell division. It doesn't want to turn on division, actually. All it wants to do is turn on the initial, the S phase, where you make all the things that you need for DNA synthesis and copy it. The cell doesn't have to divide. It's not worried about the cell dividing. It wants the cell to make all these goodies, the histones, the DNA polymerase, all the factors that are needed for DNA synthesis, the deoxynucleoside triphosphates. So it turns out that what the T antigen does is it turns on S phase in cells. Now, if your cells are pushed into S phase, and it's inappropriate, so it's not what they should be doing, it's not the proper time. If they're pushed into S phase, they tend to turn on apoptosis and say, look, you shouldn't be dividing and you better just commit suicide rather than start dividing, because dividing cells are not good news. They become benign tumors and eventually perhaps malignant tumours. And it turns out the T antigen interacts with cellular proteins to push the cell into S phase and to stop it committing apoptosis. And that's probably why in mice, which appear to have fewer safeguards than human cells, in mice it tends to cause tumours because it's causing the cells to divide indefinitely, and as I say, that's the start of a tumour. So SV40 is a little bit more independent than Parvo because it causes the cells it goes into to divide, but it has to cause them to go into S phase in order to get all its ingredients. It's assembled in the nucleus. The capsid proteins have nuclear localization signals. Histones, of course, do. And so what you get are a lot of icosahedral nuclear capsids that assemble in the nucleus. The DNA is packaged inside them, and of course the DNA is packaged with the host cell histones. And you get this huge amount in the nucleus. And one thing is, I said yesterday, the viruses tend to seem to go for quick and dirty and make a lot of quantity and don't worry too much about the quality. So it makes huge amounts of these capsid proteins in the cytoplasm. They go to the nucleus, they assemble into huge amounts of icosahedral nuclear capsids. Some of them get DNA in, some don't. And then it's got to get out of the cell. So it's in the nucleus. Why doesn't it actually go to the cytoplasm? And it seems to be that one of the reasons is we don't have a mechanism for getting DNA into the cytoplasm. We have plenty of mechanisms for getting proteins into the nucleus. So it's easier to take the capsid proteins to the nucleus and package the DNA there than it is to get the DNA to the cytoplasm. So what you get is you get a lot of these capsids piling up in the nucleus, and they form inclusion bodies. You can actually see them as things that stain for either protein or DNA in the nucleus. And so that's what the pathologist looks for, are these nuclear inclusion bodies. And so you get this vast excess of capsids, some with DNA, some without, and then eventually the cell will lyse. It's had a pretty tough time by now, and the nuclear membrane will lyse. And these nucleocapsids will be released into the plasma and be ready to go off and infect the next cell. Okay, there was a question there?
BP1, they're made in cytoplasm, right?
Yeah, the question was, is BP1 made in the cytoplasm? The answer is, once that primary transcript has gone under one of the alternative fates, the RNAs that are produced are shipped to the cytoplasm just as our messenger RNAs go to the cytoplasm. They're translated just as ours are. They look just like ours, and they're translated in the cytoplasm. And then they've got nuclear localization signals, and they're shipped back into the nucleus. So some of the features to note, I've put together the early and late messages here so you have an overview. But there are early and late functions in this virus, and as I say, early is setting the stage, and then in late you suddenly get huge amounts of DNA and you really go full tilt, huge amounts of RNA. Note the multiple use of the same DNA sequence so that you can make multiple proteins from a very small genome. And note that these proteins are often multifunctional, like the T antigen, and the host cell is providing a lot of things. The DNA synthesis, the RNA synthesis, and the histones, all of those are coming from the host cell. But in order to do that, the virus has to push the host cell into the S phase. Okay, that's a good place to take the break.